lab databases. And so I will start and then Effie will follow. Okay, next slide. Okay, so the outline.
Whoops.
The previous one. So before we begin talking about metadata and ontology, we wanted to talk to you about the status of the proposal. And then I'll talk about ontologies, and then Effie will talk about metadata and ontologies in various databases. And she will tell you about the new tool that Legume Federation is developing. And we will have some discussion at the end. Next, please. Okay, so I don't know. We couldn't remember if he let everyone know about this, but then sadly the proposal, the vaccine proposal wasn't funded. But the good thing about it was it was placed in high priority. And so there was a lot of good things that they talked about it. So they really recognized the tremendous efforts that we put in to identify common issues in the database development since 2015, right? And then they liked the goal to establish a comprehensive database with different data types for a variety of organisms that we represent. And they also liked the developing curation standard and ontologies, and they recognize it's important. The objectives of the proposals were comprehensive and clearly defined. The team are well qualified, and the proposal fits. So, but the next slide, please. The main criticism were lack of concrete benchmarking for success. So how do you evaluate the proposal was successful? And that is, I think we all think it's easy to address. We can like bump it up. And they also didn't like the lack of plan for including emerging data sets like phenomics and some new data types that we can address. But then the main thing is lack of technical innovation. So when we thought about this proposal, we were focusing on meeting, right? And like a big meeting and then discussion group, and to come up with all those standardization method and all that. But then they sort of wanted some innovative like tools or strategy that can facilitate metadata ontology data sharing by database curators or researchers, So we wanted, actually the proposal was, we wanted to assess all the available tools and then assess which one we will promote, and like we will have training sessions and all that. But then I think the reviewers felt they wanted something upfront, like some tools or some like creative strategy that they wanted to see that upfront. But then I don't know how. I mean, we all don't know how we can come up with for all these different databases and all this. I mean, we are all very like different, right? Well, anyway, so that just have that in mind, and then we can, at the end of the session, we can like discuss what we will do about it, and also you can like send us emails with suggestions and all that, So at this point, what's next? Dory has been trying to contact the program manager to find out if vaccine is really appropriate for us, like if they really want like a tool or something, if they don't want to fund like collaboration, like focuses on meetings and like discussion, right? So if we still think it's appropriate to resubmit, the resubmission is in August. And we can also submit it to RCN, but then it will be like half the budget. So that's what we are thinking. Does anybody want to add to this, Dory? Or
No, I think that pretty much captured it.
okay.
Only thing I would say is, if people can think of maybe some tools that we can add that either could be that are pretty general, maybe could be added to the AgBioData site and accessed by others. Think along those lines. I mean, we won't really know anything until I get a chance to actually speak to her. But hopefully that will be soon. I still haven't heard anything back.
Yeah, I'm curious too, because it just seemed so like such a really good match.
It did. It did.
Okay, so next slide, please. So Effie put this slide in. She wanted me to talk about definitions first before we talk about ontology and metadata. So I think everyone is familiar, but an ontology is a structured set of terms. That is, terms have relationships to each other. A controlled vocabulary could be viewed as a very simple ontology. Typically has no structure, just a list of terms. Okay, metadata is data that describes data. The structure of the metadata is defined by a controlled vocabulary or fields. Many values in those fields consist of ontology terms. Formal metadata schema and ontologies are necessary for find, describing, and comparing data across multiple data sets. Okay, next slide, please. So I just want to briefly talk about ontology, curation in GDR and CottonGEN, CSFL, CGD, and Vaccinium, I think. Okay, so mainly we do Go association for gene models and transcripts. And the second thing we do is associating trait ontology and developing trait ontology if it's not available for QTL and breeding data, cultivar evaluation data sets. Next, please. So Go assignment to gene models and transcripts, I think almost everyone else does this similarly. So gene models and transcripts are associated with Go and Interpro domains using Interpro scan. And we use this associated terms in searching and downloading. On the right, it's a brand new module, triple module that we developed. So here you can search any kind of data types. And one of the category is the functional annotation that includes code terms and CAG and Interpro. And then on the right side, users can check whatever field, whatever data that they want to download about this gene or transcript. So the functional annotation term, including Go term and accession, are included. Next page, please. And similarly, we do trait ontology assignment to the trait terms for QTL and cultivar evaluation data sets. And what we do, it's a manual curation, and what we do is we look for appropriate plant trait ontology terms first and assign them. And then if we don't find it, we create following like the customs of trait ontology, we create new terms. And so we have, for each database, we have rosaceae trait ontology and cotton, citrus, and cool season food legumes trait ontology, and vaccinium trait ontology. And each database lists this. And then we submit them, but then it's a long process for TO to accept them. So, and then similarly, we use these trait ontology terms to search and download the same module showing here. And so we associate, like they can search it by the trait root terms, TO root term, which we just say trait category, and the actual trait name. And then they can download, check to download on the right side. Next, please. And for cotton, our cotton curator, Jing Yu, does a little more work on manual curation. So So she associates the trait descriptor found in QTL papers with trait terms as well. I mean, with trait descriptors as well as trait terms, and the descriptors are similar to crop ontology terms because it has units and the method. So I think she's trying to meet up with them to submit these to CO as well as TO. So depending on how much curator time we have, we could do similar things for our other databases. Next, please. And then one other thing we've been doing, and we kind of like need to redo, like gain this effort, but then so what we did was we met up with crop ontology people and then trait ontology people, and we submitted rosaceae terms, and then crop ontology team is trying to assign POEC and KEBI or NCBI taxonomy terms based on our trait ontology. You can do that because trait ontology is often associated with plant anatomical terms or enzyme or chemicals or taxonomy terms plus the trait. So I think she's using both scripts and manual curation. So this was one of the things I think we said in the proposal, that this could be developed as a more automatic tool with less manual work. Okay, so I think this is the last slide for me.
Okay, I will take over and try to get through some of this pretty quickly so we can have time to discuss. So starting with MaizeGDB. At MaizeGDB, we have focused very hard on genome metadata. We're following the MIXIS and its plant extension standards. I was part of the Genome Standards Consortium as it developed the plant extension. But although a lot of what I did was just listen. So we created some metadata templates, and these were served a dual purpose. One was to collect all the information for GenBank or the European Nucleotide Archive submissions for genomes. And then the other was to meet the MIXIS and the plant extension recommendations. And then we added some MaizeGDB specific fields like URLs to browsers and to downloads and so forth. And then the resulting data templates are usually filled in by data providers, but even when we get pretty complete data back, there's quite a bit of time required by a data curator to make sure it's all correct, specified correctly and accurate and so on. So it takes doing. Even a single genome can take a few weeks to get all of the metadata together. So once we get it and we're allowed to show it, we split it up a lot like it shows up the data schema at GenBank. So we have the project information, and then sample information, then the sequencing and assembly information. And then we add assembly statistics. And the Genome Consortium Standards Group was planning to address this, but decided it was too soon and that they didn't have people in the group that were sufficiently expert on these things. So at MaizeGDB, we sort of made up our own standards for statistics based on what's most common. I hope that eventually, or maybe soon, that we can get a recommendation for how to gather and display assembly statistics so that people can compare across different assemblies. And then we also have metadata for annotation. This we had to make up because apparently there isn't any. I went out to Fair Sharing and asked. Fair Sharing gave me a bunch of standards with annotation in the word, but none of them were for the structural gene model annotation. So overall, we're doing a pretty decent job with the genome data, metadata. We're doing what we can with the annotation metadata. As far as ontologies go, MaizeGDB has been on the scene before ontologies are really a force to be reckoned with, so it has lots of legacy terms. But we do bring in the Go ontology and plant ontology terms as we're able, and other ontologies. I hope that we're trying to improve, and where we find ourselves needing new terms, look to make sure that we're not duplicating one that already exists. And then other types of data, aside from the genomes and the annotation, we don't do so well. They're generally described by reference only. And then the metadata about it can be augmented somewhat by contextual data. We can stick functional annotation of the gene models and then list the reference. We've not, for example, attempted to apply Miami to expression data, but on the other hand, that's a really big job, and we don't really have a lot of expression data at MaizeGDB. We are starting to address this metadata problem with our bulk download files using README files. And I'm just going to stick these up there because these are worth exploring, but I won't talk about them much here. But here are two really good examples of the use of genome metadata. Patrick, this is microorganisms that has, I suspect, hundreds of genomes that it maintains, and then the Genomes Online database that's sort of a global genome database that gives information about every genome assembly that it could locate. Okay, PeanutBase, not so good. There is genome metadata. It's not as good as at MaizeGDB, but it's there. There is no annotation metadata. Ontology is a little bit better, as we started using these right from the get-go, using soy, plant ontology, and Go. The peanut type terms are hard to find in the plant ontology, and hence the reason for the soy. But we pretty much had to create our own terms. But then these are correlated back to formal ontologies. Other types of data, we've got germplasm, trait data, QTL markers, functional annotation, gene expression atlases. And of all of these, except for the QTL data, none of them have metadata. And I was quite dismayed to realize this. There is some study data that's associated with the traits, but it's not in a good form for using, but not terribly useful in its current form. So hoping to address this. So I've got this new data coming in. I'm working with crop ontology to get all of the peanut-based traits into the groundnut ontology. This is CO3337. And then I want to correlate the GRIN descriptors to both the trait ontology, plant trait ontology, and to the groundnut ontology. And when that's done, Plantium will take that in. I'm getting a lot more trait data. This means I need to do a better job of study metadata. We're going to be bringing in pre-breeding germplasm collections, and we expect a fair bit of genotype data pretty soon here. So some of the approaches, we're going to adopt the breeding API, pretty close, I think, to making this real. This is developed by Valentine at Bioversity, a triple module. And I'm going to see what it will take to implement the minimum information about a genotyping experiment. And then also, I haven't done the research yet, but I suspect that there's a standard for describing trait studies. Okay, LIS. There is no metadata collected for the genomes and annotation in the website database. I just have not had time to do this. And the data presented on the website, the only metadata that comes with it is, for the most part, just the publication. However, a lot of work has been put into the Legume Federation data store and a tool called DS Sensor. And this is to focus on collecting, managing, and searching the metadata, and also provides a means of attaching metadata to files. So I'll give you an overview of this. It's in GitHub, so I've got the link up there. So the full data sets are provided as downloadable files. These are not custom bulk downloads, but these are the full data sets. They're given strictly defined identifiers. Each data set's given a unique identifier, and this is part of the file name. And so this means, there's a key that you can use to get the metadata if it should go astray from the file. We have very rigorous directory structure and directory file naming rules. The metadata itself is in both human and machine readable files using YAML. And then we apply a very simple hierarchical web interface that has some additional capabilities, but people are used to using file browsers, so we decided this was a lot better than trying to get fancy. Important point is this is not a permanent data repository. It is mirroring, for the most part, mirroring data from other sources, but it puts it all into one place in a consistent way so people can get to it more easily, including the websites, because a lot of the data in the data store are read into, for example, CMAP, or it's also used to populate the Legume mines. So the directory file names, what it amounts to a controlled vocabulary, starts with organism, cultivar accession, the data type, its version, its key, what's in the file, and its extension. And we use what we call a GENSP abbreviation. This is what's used by Uniprot. Use 3 letters of the genus and 2 letters of the species, typically the first 3 and first 2. Sometimes you can't. You have to get a little creative. So Arachis hypogea, this is cultivated peanut, and that equals arahi. So we have this file called arahi.tifrunner.genome1.kyb3.genomemain.fna. This is the FASTA for the genome for the tifrunner, first version of the tifrunner assembly. So we have a set of names that we use to indicate the data set types. For example, GNM, genome, annotation, MRK, markers, map is a map. The file types, genome main, genetics, CES, protein, there are quite a few of these. And then FNA would be a nucleotide FASTA, FAA is an amino acid FASTA, CMAP, GFF, so on, several of these. Okay, the metadata inside each of these data store directories. There's 4 files, and these are all checked into the Legume Federation data store, which tracks changes to these files. The main one is the README, and this describes the data set itself. There we have 26 defined fields. The descriptions, this lists all the files of the data set and what they hold is a very brief description. Correspondence, this lists any changes that were made to original files from the original source, and then a checksum, so people can verify that they got everything when they do a download. The interface looks like this. Looks a lot like an FTP browser, but with some additional features. This is implemented with H5AI. So you can do things like you can decorate it, put information at the bottom. So here we describe the abbreviations and some information below that about how to use this. At the top is an overview of what's in the directory, and this is built directly from the metadata in each of the directories. Here's the inside of one of the folders, continuing the tifrunner assembly example. You can see what the names look like. This README, if you double click the README, it'll come up in the browser, and it has this information, where the data came from, what it's about, publication, a description of the data, dates, how the files were about, transformations and changes that were made to this data set. So you can do a few things in this FTP looking thing, H5AI. So if you click this hamburger menu, you can change the appearance of the listing. And then, if you remember, part of the reason for the key is to attach the metadata to the file. So say I've got this file someone gave me, and I don't know what it is, but it has this key, 6LNX. So I can come out here and type 6LNX and say search, and I find, oh, this is Lupinus albus annotation file. So I can come down to the manifest descriptions and find out exactly what this one file is about. I can see the other files in the group, and I can check the README file, learn more about the data set as a whole. Okay, so this one instance of the data store, the Legume Federation data store, is being used by 4 resources: Soybase, PeanutBase, Legume Info, that's LIS, and the LegumeFederation.org. So one of the problems, though, with this, so it looks pretty clean on the surface, but it's really difficult to prepare a data set. It takes a ridiculous amount of time. There's a case where we put the simplicity on the front end and the complexity on the back end. And because it's being done by humans, it's, there are typically errors after a set's been prepared. So Connor and Andrew at NCGR got together and got this idea of creating a tool called DS Sensor, and then Connor ran with it. There is it's in very preliminary stages, but you can get the code from GitHub. And for the time being, it's mainly an administration tool, but it does show some promise. So it's being useful for researchers to explore the data. Backend is Python, and it uses a graph database, Neo4j. It provides an extensive web service API, so other tools can query this thing to get information about the data store. There's a JavaScript web interface that gives overview of what's in the data store. And then there are 2 command line tools. And I was hoping to tell you all about the web service API, but I was unable to get the information from Connor. I will add it to the presentation before I hand it over to Jacqueline to be archived. So it will come. That information will show up here. The web interface, this is active. Again, this is in very early stages, but you can go out and play with this. And there's the URL. So it gives you overall views of what's out there. Here's a circle graph of all of the genome assembly data that's in the data store. This can be filtered by source, like LIS or Soybase or whatever, or by genus and species, so that you can get an overview of what's out there. The tool also does some analysis of, for example, the genomes. This is a fairly long table. I'm just showing the very beginning of it. But it goes out, checks all these assemblies that are in the data store, and it computes things like the BOOSCO and the N50 and the L50 and so on, various standard measures. So the command line tool is a normalizer. Okay, this is one that's trying to help get these things into the data store. So you can give it a dataset type, a genus, a species, a subspecies, or it might be a cultivar or an accession. And it will create a directory with a set of files that you need for that data type. It will create a README for that data type with all the fields set and ready to be filled in. And depending on the data type, it may do, and what you give it initially, it may rename some of the files to meet the specifications of the data store, and it may also go into those files and change identifiers, for example, chromosome names. Although it's careful to preserve the original names, so it's these identifiers are typically changed by adding a prefix rather than changing it completely. The detector, you can give this a directory, a genus species, or a specific data set. It'll go out and check, make sure all the files are right, all the names are correct. It'll verify that all the fields are in the README. It's not yet checking values, but that's could be done. Verifies that the file formats are correct for, for example, running a GFF verification on GF files and so on. And then if requested to do so, and if all the tests pass, it will add the data to the Neo4j database, and then it will be available for the web browser that I showed earlier. Okay, so this was an interesting presentation to get together because I realized I'd fallen, for all that I talk about metadata, I've fallen pretty short of doing a good job with it. So this is where we get into discussion, and why is it so difficult to do this? And I think probably the key is it takes a lot of time. It is difficult and it takes a lot of time. And none of us have an abundance of time on our hands. I think the situation is improving, but we still, I think, have problems with data providers not understanding the importance of metadata or of using standard ontologies, or they assume that the data curators will do this work for them. And then the recommendations are complex. So it was startlingly difficult to make those data templates for MaizeGDB, even though we had the standards sitting right there, and I'd helped with the plant extension. It still was quite a challenge to figure out what parts of those to put into our data template. And even there, we found that there were things we needed that weren't in either of those recommendations. And then using standard ontologies, and so can mention the difficulty of getting these terms, new terms into things like the plant trait ontology. We've been trying to get terms into the crop ontology for about 3 years. I think we're close, but it is difficult to do this, and so it slows you down. And we all have too much to do. We can't be sitting around twiddling our thumbs. So this is another thing I think that makes using the standards, the ontologies, difficult. And then the other discussion point is what's better. There's the DS sensor that tries to help with this. There's a 2019 paper on the Breeders API that points out that using a standard API and data schema and data exchange format kind of helps you define and enforce and federate metadata. And then I haven't had time to look at this much, but there is a cross-discipline effort at working with metadata called Metadig. I glanced at it. It wasn't immediately obvious there's much there that would apply to the kind of data we work with, but I thought it could bear some additional study, as it's trying to be a fairly generic approach to this. So we have close to a half hour for discussion. So I'm opening up the floor.
Hey, Effie, can you hear me?
Yes.
Okay. Yeah, I had so many, I nodded my head a lot of times while you were talking. I think the DS sensor application looks great, and it kind of speaks to some of the problems you were bringing up in the end of metadata is hard. Why is it hard? It takes time. Why does it take time? Because the standards are loose and there's a lack of technology for those loose standards and actually having the curators implement it, I guess, and this is one of the things that BRAPI is trying to address. So I had a number of different questions, but maybe I'll just start with kind of a random one here. iRods, so do you know about iRods?
Yes.
So that's another, because we've been having the same problems with the metadata associated with flat files. And how do you structure it? Because there's not really a technology that we had available at the time to put things up, except put it on an FTP site with a README file, right? And I really like what you guys have been doing, but we'd explored iRods initially and kind of rejected it because it's former technology. Have you looked into it, and do you have any thoughts about that? Because I know that's what fibers.
Yes, that was the first thing we looked at when we started. When this was one of the data store was one of the fundamental goals of the Legume Federation project, and we started with iRods. And also decided it just wasn't going to meet the needs. I can't remember all of the reasons, but it's a little tricky to work with. We would have had to create some, a buffer to spare the user from some of the complexity. But it also didn't meet all it was, I can't remember if it was just insufficiently flexible or too flexible.
Right.
Um,
Right.
But yes, we did try to make iRods work for us and decided.
And it's another big tech stack, right? So you have to maintain it and set it up and whatnot.
Right. That was another issue. Yes.
Yeah. Yeah. In terms of the bigger questions for the grants too, what I really struggle with is to define concretely the problem around what metadata as a larger group we need. It would be great if we could do something like BRAPI, where it's like, this is what we're doing, this is the standard, here's the technology. And if AgBioData could identify a few data types where this metadata problem is still problematic, despite the Genomics Standards Consortium's work and other triple work or whatever, maybe key in on that and identify what can we all do the same way and support a technology that implements that. I mean, that might be one of the tools that we could work on, but I still have a hard time wrapping my head around whether or not that's possible to define if we can all agree on the same set of metadata and what types that would be. You know what I mean?
Yeah.
Because even with the genomic, I mean, you laid out pretty clearly for the genomic data, you were able to do that with work for how many genomes? Like 10 or so for peanut and maize.
Yeah.
And that seems like, yeah, that might be a straightforward one where even we might even still be having some problems about the genome metadata and maybe the metadata for the samples that were used to generate the reads.
Yeah, I'm really curious how Patrick does it because they have hundreds.
Yeah.
And the metadata looks pretty thorough to me.
Yeah. And NCBI might be another partner there. I'm sorry, go ahead.
No, I'm just wondering about the the interaction with the community. Can we work with the community in a way to make it easier to get metadata and to get all the boxes filled on the front end? Would that help with any of this?
It helps, but it still takes the curator to go through and check it.
Yeah.
Yeah, they could be, we could automate some of this, for example, verifying that the accessions are correct and so forth. But it's hard to automate things like, did they enter the right value for the sequencing technology? Did they get the correct, did they spell all the words right? It's one.
Yeah.
Yeah,
we'll have to see with oh, sorry, go ahead.
I was just going to follow up, and so the BRAPI, would it make sense for us to say, well, we're going to do this with, say, phenotype data? The BRAPI is, I think, a really, really successful project, and it's also a really big one that's been around for years. They have hackathons. It's a very there's a group dedicated to it. It's not something that you do along with something else. So I think that might be too big to try to tackle with AgBioData at that scale. But I also like the idea of coming, saying, okay, we need something like this for phenotype. Here's what something's been started. Here's some starts.
Right, and given that BRAPI already exists and there's a track record for it, the grant reviewers might be familiar with it. Perhaps we could still argue we're trying to pave the way towards an end goal like this, but before we can actually do that, we need to, the point of AgBioData is to identify the standards and the problems and the data types, and then that would be the follow-on for another grant that would be development-based.
Yes.
Yeah.
Right, yes, exactly.
Yeah, and if we could get that conversation started with the program manager to understand what they're really looking for, or what that particular review panel were looking for, that would help us answer some of these questions, I think, of where we go from here.
I think I had one or two more nerdy detail questions if there's no other big picture questions right now. Oh wait, Jackie had one. Brappy might be the speaker for the October conference call. Okay, we'll see. You had the metadata for Legume IS up in GitHub. Did you also have the data up there?
No. Yeah, I should have mentioned that. Yeah, only the metadata files are being tracked.
Okay.
The files themselves are too big.
Okay, yeah, we'd considered that for some of our data files too for the version control, but we'd thought about playing with Git Annex, but that kind of fizzled out.
Yeah, another aspect of this, the data store in general, and managing data sets like that, is tracking changes. If you need to change identifiers, or you run a new analysis because you found that the file that's up there was incorrect, so you have to fix it. So using GitHub was one way to track changes to a data set, but through the metadata rather than the files themselves.
My next question was, sorry, I'm just dominating here. Do you know what your users use the genome assembly metadata for? Or if they use it?
I suspect it's not used much yet. DS Sensor is in its infancy. My hope is, and I wish I could have shown the API because it's pretty neat what you can do, but my hope is that, for example, cold season food legume could use the API to pull data out of the data store and use it. I don't know how close we are to doing this or how committed we are, but there was a lot of talk of putting genome tracks in there, and so that could be used to feed genome tracks into web genome browsers at other websites.
Right.
The CMAP files can be pulled into the new CMAP.js app from the data store. You can do direct links into directories or data sets or specific files.
Yeah, Effie, all of the Metacargo HapMap.j browsers are being driven off the files in the data store.
Ah, that's good to know. Good.
And isn't Sam also loading mines?
He is beginning the effort of loading mines from the data store rather than from the Chado. But yeah, that's basically the idea, is that by kind of centralizing where all of the downstream tools are getting their data from, there will be greater consistency among the representations in those various systems.
So some things we've learned along the way in making these data store collections, to make it machine usable, some elements of the standardization have been really important, like for assemblies and annotations, coming up with a name that indicates that this is the version to use. So we have a genome main and gene models main. So if a machine sees those are the versions to use in a browser. And then in the README, there are some fields that are used by scripts. For example, the overview for a whole collection is composed by reading the subject field from each of the contained READMEs, and that goes up into the header.
Very cool. I asked about what they use because of the other question, so that researchers are often reluctant to contribute good metadata because they don't often see the point. And I've been trying to work with this too, because we all have that same observation, right? And part of it might be that, do we have a good use case where we can illustrate to the user, to reviewers, like this is the magic that can happen with complete metadata. These are the studies that you can perform. Is there such an example out there?
If it's a genome data, I mean, if it comes from like different cultivars from the same species, and if they specify it, like, you know, and also users could pull it out and do a synteny or something. And like if you do expression data, the metadata will be really important. And I think users can pull out like the expression data from different stage or whatever tissues, and I think the use case would be, I don't know if they do it right now, but then in the future it will be really useful.
Right, that's the problem. It's like you convince people right now, or like saying like, why would I bother telling you that I have Illumina?
And we can show them like this is
whatever.
Yeah.
Well, I think there are some examples. There's Animal QTL and Patrick, and the Gold Genomes Online database.
I think we're not doing enough outreach to the researchers. I think that, and I sort of see watching this happen in MaizeGDB. You know, originally we didn't, you know, really know that much about metadata, and we took these sequences, and then people would ask us, well, you know, how was this assembled? And oh, we better go ask them, and blah blah blah. So, and I remember, yeah, you know, arguing with people back and forth whether they need to fill in this one stupid box or not. So that took a lot of time. But now it's like, now that we get more assemblies and we talk to the people more that are doing them, we just say you have to do this, period, or we're not going to post it. And I know that that's sort of hard line, but then when that becomes the norm, people will be able to, I don't even know if giving people examples will help until they have their own personal examples.
Yeah, you might be right. But yeah,
I mean, nobody wanted to put their sequences in GenBank when GenBank first started, and then now it's a no-brainer. You know, of course, everybody sees the use of it.
Yeah.
That's not a very good answer, just forcing people to do it. But I think that's...
No, it might be worth still doing is just thinking out like a really, if we're going on the recruitment trail, as you're saying, Lisa, to come up with a good slide, a good use case of this is why you want to give us your metadata. And
I gave this,
what you can do.
Right? I
should
do a report of this sometime, but I gave a talk at the Maize meeting to the researchers on 8 simple things you can do to make your data fair. And they allowed me to give it in the regular session, not in a special community session that nobody attends, And man, I got a lot of people didn't know those things. I got a lot of discussion afterwards while we were drinking later, that, oh gosh, I want to be a good data citizen. I want to be able to do this. I didn't know I should do this. I was really surprised. I'm so deep into the database thing that I just forgot these people just don't even know. They don't even know about this. They don't know about fair data. If you say fair, they don't have any idea what that is. So I don't know. I think that interaction.
Can you share that?
Could I share that?
Yeah, the talk.
Absolutely. Absolutely. Yeah, absolutely. We can put it on the webpage.
Yeah. I see here, Megan, you're a librarian, right? The other thought is there are people who are professionally trained as metadata librarians, and that their bread and butter. Most of us here are probably trained as biologists who came into it kind of from the other end, but maybe we can have more conversations with librarians too about, is there a framework to convince people from the other end?
Yeah,
this is Doreen. I don't think there is, because I've been trying to do it for a long time.
Yeah, bummer. Okay.
There is more momentum gathering. This is Megan. This year, though, than in past years, with some high-level groups for universities, at least. So that's good news.
Yeah, some of the disciplines, like the geologists, a lot of the environmental science, they already know what metadata is. But the average researcher I talk to, it's what are you talking about?
Yeah, they do it all the time. You know, they provide publications are basically metadata. You know?
Right.
Everything you need is in the publication.
You know, this simple thing is that people told me afterwards they didn't understand, they never thought about what machine readable means.
Oh, really?
Yeah, I mean, very basic, simple things like, oh yeah, this an image of a FASTA file is not the same as a FASTA file. Oh, I never thought about that. I think they're way behind what I thought they were at. And I think just understanding that makes them. I don't think they're trying to resist. I think that it's something we can. I don't know. They knew about it. I think the obviousness of why it's good to do follows.
One context that I think we've found is driving keener interest in metadata among our users is comparative applications where you would like to be able to draw conclusions from taking genomes that have been produced by different groups and getting some biological insight into it. But if they're all being produced using different technologies, you have to kind of temper your conclusions with some level of skepticism about, are you going to get the same information out of a PacBio assembly versus a 100 Exolumina assembly? And so some of the reason why we developed some of the aspects of DS Sensor was to allow users, for example, to say, okay, well, you've got 20 Medicago genomes in your system now. Can I quickly look at the assembly stats over all of them and see, are there outliers and things like that?
In our selection of fields for the metadata in the data store, we looked at a number of different metadata collections from libraries like the Darwin Core to GenBank to a number of others, and we tried to keep it minimal. 26 fields is pretty slim. A lot of the fields were really for our purpose for tracking data. Where did the data come from? Does it have a citation? Were any transformations done on the data? What was the previous file name? Things that we have to maintain as data curators ourselves.
Yeah.
I have a question, this is Megan, about a slightly different topic. If we get some of this genomic data that's going into places like MaizeGDB, and we're supposed to be the long-term host, is there a way to get the metadata you have generated about it into our system?
Well, I think if MaizeGDB were using DS Sensor, the answer would be yes.
Okay.
It's probably yes anyway, but it would take more handwork.
Yeah, that's what I was afraid of. So I'm not 100. It's only happened like once so far that I know of, but may start happening more often.
Yeah, it's a good question. The handwork could be a link to, the metadata page for the genome in question. But would it be that difficult to offer it up as a web service and you could just query it and grab it?
Mm-hmm.
So that's.
I'd vote for the web service.
Yeah, I'm very fond of web services too.
Well, and then as the archive, should we just save it as a plain text file and include it with the download? Is where I'm coming from at the archive level.
Right.
Because you guys do a lot of metadata work and it's great, but I also don't want it to get lost.
Yes, good point. Can you point me to your example?
Yes, though I don't know if it's in MaizeGDB yet. So if it's not, don't get mad.
No, it's okay.
I have to find what I think.
They jump all over the researcher.
Yeah, I'll put it in the chat.
Okay.
I think this is the one that we suspect might come to you.
All right. Yeah, this is a familiar project.
So something, yeah, so something to think about on my end is like integration, because you guys have a great in-system database, but ours goes into things that are more generic, and trying to get everything to talk to each other is an ongoing
Right,
challenge.
so it might be something like the MetaDig might be useful. But again, I haven't looked at it hard enough to see if it is or not.
So Abby, I'll follow up with you on that one later.
Okay, sounds good. Have any more comments? We have 3 minutes.
I love ontologies. That's a comment. I wish we would use them more and all the time.
I will say that I volunteered to Connor, and he was happy. I mean, happy. I don't know how happy he was. He was willing. I think we will schedule a time for him to talk at least to people who are doing Legume databases, but I think maybe we'd advertise it through AgBioData so that anyone who wishes could get a more in-depth tour of the DS sensor.
That'd be great.
I barely touched the surface of it. I'm sorry?
Oh, I just said that would be great.
Okay. Anything else?
I think that's well, anything from the people that didn't say anything? A lot of people are still muted.
Yeah. Well, if you have questions or you have thoughts, send emails.
Thank you very much, Ethy and Sook. That was great.
Hey.
Thank
you, folks.
For coming.
Thank you.
Bye-bye.
Bye.
Bye.
Bye, everybody.
Have a good week.